Yeah, it's my real pleasure here today to introduce our speaker, Jason Price. Jason did his PhD at Washington State University, where he studied the role of antimicrobial peptides on multi-drug resistant gram-negative bacteria. Since then, he worked with Dr. Jim Olson here at SCRI, where he was very productive and published several high-impact studies. He's a protein engineer specializing in novel therapeutic design for the treatment of pediatric cancers. His work sits at the intersection of protein engineering and translational therapeutics, using engineered proteins as precision tools to change biology in ways that individual antibodies or small molecules may not be able to. He started his lab in July of 2024, and I think today he's going to talk to us about antigen assimilating multispecific T-cell engagers. So please join me in welcoming Jason. Thank you.
Thank you for the very nice introduction. And thank you, Sean and Rich, for the invitation to speak here today. And thank you all for your attendance. So I'll share some stories today from our lab. A lot of this is technology that I developed while a postdoc in Jim Olson's lab. And then now as I've started my own group, have continued to advance some of these ideas and concepts. And I'll say I'm a protein engineer first. And it just so happens that we build some designs that engage the immune system, but I'm not claiming to be an immunologist. So just take it easy on me in some of the questions. So the outline for what I'll talk about today, I'll give an introduction to this unique class of therapeutics, which we term T-cell engagers. I'll introduce the concept of antigen assimilation. And then I'll sort of touch on how we're attempting to build best-in-class multi-specific T-cell engagers that take into account these concepts for DMG. And I'll touch on some of our advancements with dual antigen selectivity, how we are attempting to find new antigen assimilation targets. I'll touch on some novel CD3 binders that we've discovered and why they're unique. And then I'll also hopefully have time to touch on some of our co-stimulation platforms that we term synergistic adapter molecules that we think can help unlock more activity of this class of therapeutic. So I'm sure most people in the audience are aware of T-cell engagers, but just to give a brief introduction, T-cell engagers are protein therapeutics that are more or less bispecific antibodies or bispecific binding domains. One arm binds to a tumor cell antigen, and the other arm binds to a T cell. This more or less links the T cell or directs the T cell to kill the tumor cell. And although shown here in the cartoon is this direct phenomena where you have one molecule that's linking a T cell and a tumor cell, we really think about these a little bit different than that, in that these are really therapeutics that allow for T cell activation locally. So through the tumor, engaging with the tumor antigen and engaging CD3, we're able to activate T cells locally where the tumor antigen is present. And so it's more than just simply directing this T cell to kill this tumor cell, but there's also lots of cytokine release, lots of other factors that happen when this phenomena occurs. So this is a list of the current FDA approved T cell engagers. One thing that really stands out in this is that almost all of them are against B cell targets and for B cell disease. That's because these drugs work phenomenally in that setting. What you'll notice is there is now one FDA approved drug, tarlatamab, for solid tumors. But really there's something fundamentally different between B cell disorders and solid tumors, and really figuring out what it is, why these things work so great for liquid tumors but not for solid tumors, and what can we do to address these hurdles that are found in solid tumors but not in liquid tumors, is really what we're focused on in the group. So to say that again, here's a list of challenges for translating the success in liquid tumors to solid tumors. So some of these are shared, but many of them are unique. So tumor selectivity, off-tumor toxicity is a big thing. So when we think about antigens that we can target on solid tumors, some of the best ones like HER2, EGFR, these are things that are also expressed on normal tissue. So when you have a potent drug that can target that antigen, you need to be aware that there's a tox risk involved in going after that. Tumor heterogeneity is a giant problem, where most of these tumors, once again, HER2 EGFR is an example, and they may be present on 50% of the tumor cells, but in order to have a successful therapy, you need to eliminate 100% of the tumor. So tumor heterogeneity is a giant issue. Part of that is antigen escape, or even if you have a good antigen, tumors can find ways to downregulate that target or escape targeting it. Immune checkpoints, we're all aware of PD-L1, CTLA-4, and others. These are targets or ways that a tumor can express on antigen on its cell surface that can shut down T cells. Immune dysfunction in the tumor microenvironment. This is things like certain tumors making TGF-beta, or in general, tumor microenvironments being immunosuppressive from a mixture of factors. And then finally, maybe the biggest issue that T-cell engagers face is unnecessary or non-productive cytokine release. So almost all T-cell engagers run up against toxic cytokine release or ICANS in the brain. And this is really makes it difficult to dose T-cell engagers into a range where you can find efficacy, because you often hit CRS dose-limiting toxicities first. So, it looks not too bad. These things all seem achievable. But the first ones I'll talk about with our antigen assimilation concept is that we think we can address tumor heterogeneity, antigen escape, and immune checkpoints with this idea. So I just wanted to introduce DMG, diffuse midline glioma, real quick. Being a postdoc in Jim's lab, this was a large focus of his group, and it's a really tough tumor to treat. Maybe if we're looking to try to conquer solid tumors, this is the hardest solid tumor to try to go after, but that's in part why we're interested in it. This is a really devastating disease that's more or less universally fatal. It has a few mutations, it's unresectable, it has poor response to standard of care, and although there's a lot of promising work with cell therapies, including those here led by Nick Batanza, the reality is that there's a lot more to do here. So we're using this as both a target, although what we're building is can translate to other tumor types. We're using this as the model we're going after first. It's also a great model to show just the problem of heterogeneity. So this is some single cell sequencing data set that really highlights this issue. So these were 6 pediatric patients with DMG. Their tumors were evaluated through sequencing at the single cell level, and they were able to separate malignant tissue from other immune cell types. And then what you can do is you can then ask questions like, okay, how heterogeneous are these targets that we're going after to try to treat this disease, like EGFR, HER2, IL-13 receptor alpha, B7H3. And what you can appreciate is that across patients, there's a lot of difference in the expression of these antigens. And then even within patients, blue representing a high level of expression, yellow representing a low level of expression, that you see a lot of yellow on these maps. So I think this is in comparison to something like beta-actin, just to show this is what a uniform expression of a target looks like. So I think we can appreciate that heterogeneity in this tumor is really an issue. The other sort of introductory point that I wanted to make is that this immune checkpoint PD-L1 rather phenomenally is universally upregulated in response to interferon gamma. So this is a universal phenomena of tumor biology. We observe it with pediatric brain tumors. We're showing this here by flow cytometry, where we're looking at PD-L1 expression levels on the cell surface. And we can see that many of the tumors that express some level of PD-L1, that's in the turquoise, when you add interferon gamma 48 hours later and stain again, you can see a significant upregulation of that target. What you can also notice is that some of these pediatric brain tumors that don't express PD-L1 at baseline, when exposed to interferon gamma, then now put a lot of it on its cell surface. So even if you don't have any PD-L1 at the beginning, after interferon gamma exposure, you upregulate it. And this isn't just a phenomena that's found in brain tumors, but it's also found in many adult solid tumors. We're just showing here lung, breast, colorectal, and glioblastomas. So really a universal phenomena that PD-L1 is upregulated when interferon gamma is present. So that then leads us to this design of our antigen assimilating idea. So we have here, this is just a FC scaffold. We have VHHs that target a tumor selective antigen. We have T-cell engagement so that we can bring in the immune system. And we've also now included a immune checkpoint blockade molecule against PD-L1. So in the case of this proof of concept design, we're using ROR1s that were discovered in-house from a camelid VHH discovery campaign. We're using traditional CD3 from the literature, and we have a PD-L1 molecule that was discovered by Zachary Crook, who's in the audience here, when he was a postdoc in Jim Olson's lab. And then so to sort of highlight the idea here, this is an example of a heterogeneous tumor where you have some ROR1 positive cells and some not ROR1 positive cells. You can imagine that if we built a T-cell engager that just targets ROR1, we're going to be able to eliminate these target positive cells in the best case scenario, but the target negative cells will be unaffected. They'll grow out and we'll get persistent tumor growth. What we imagine with our antigen assimilation designs is that if we target the ROR1 positive tumors, that creates an initial spark of immune activity that releases interferon gamma from the T cells. So when T cells are activated, they release interferon gamma. What we then anticipate is that the tumor cells will respond to that and upregulate PD-L1. So this is, we kill the ROR1 tumor cells, T cells come in, release interferon gamma. That creates cells that were originally PD-L1 negative will now upregulate it on its surface. Because we've incorporated a PD-L1 targeting arm into our design, we can now anchor down on that and kill the cells through PD-L1 CD3 axis. So that now, once we kill ROR1 cells, we convert everything to PD-L1, which we can now target. So that's this concept that we're calling antigen assimilation. So we thought, how can we best demonstrate that this is a real phenomena? So we engineered a co-culture system here. We have ZR75 cell culture line, which is a breast cancer cell line. It's PD-L1 negative and ROR1 negative at baseline. We then also engineered a ROR1 overexpression cell line that puts native levels of ROR1 on a cell surface. And we created this 10% engineered co-culture system. So now in the dish, we have 10% of the cells are ROR1 positive and 90% are not. We then produced a PD-L1 CD3 BiTE and asked, is it able to do anything in this setting? And as you would expect, that we're looking at an IncuCyte T-cell killing assay where we put tumor cell co-culture in the dish, add T cells, add our drug, and we're looking at tumor cell growth over time. And what we observed, rather expected, because there's no PD-L1 present on either of these cell types in the co-culture, when we add BiTE versus the PBS control, we see no difference, no significant difference in tumor cell growth, or no elimination of tumor cell growth. Everything grows out just as you would expect. There's no killing. So now when we add just a ROR1 CD3 BiTE in this 10% co-culture model, now we would anticipate to kill the 10% ROR1 positive cells where we're unable to affect the negative cells. We would anticipate that we would get persistent tumor growth. And that's exactly what we see. Actually, very interestingly, is we see a negative effect of adding the T-cell engager, such that once we've killed the 10% of the ROR1 positive cells, we've actually angered the non-ROR1 positive cells, and now they grow faster. And this is a very true phenomenon. We see it repeatedly across models, across T-cell engagers, that tumor cells respond to cytokines like interferon gamma, TNF alpha, and other things, and they will grow faster in response to those signals. So we've just sort of pissed off this tumor by killing a little bit of it. It now responds and grows faster, and that's why we see significantly this red line above the gray line. So then we're very excited to see that with our antigen assimilation proof of concept molecule, that now we can kill the 10% ROR1 positive cells. That will then convert the rest of them to PD-L1 positive cells. We can then use that axis to eliminate the entirety of the heterogeneous tumor. And that's precisely what we see, where at early time points, we actually anger the tumor because of the cytokine release. But then because the PD-L1 expression, we're able to then target that and completely eliminate tumors in the well. So this was our first proof of concept that that we can actually do this. We've done some other experiments now to show just exactly how much of the tumor needs to be antigen positive for us to achieve this phenomena. And so here we did mixtures of 50-50, 25-10, 5%, all the way down to nothing. And you can see that if we don't seed the culture with any antigen cells, we don't have an effect. And then based on the level of antigen positive cells in the mixture, we have a increased kinetic and increased killing, but in all cases, as low as 5%, we're able to sort of create this phenomena, turn the tide, and begin to kill the antigen negative cells. We did this just mechanistic. It's rare that we do this, but we did in this case, where we proved that this is interferon gamma dependent. So here we have this co-culture. We're looking at the ZR75 target negative cells in the 10% co-culture. This is the tradition. We're now, rather than looking at tumor cell growth, we've inverted these curves. We're looking at percent T cell killing activity. So 100% activity means 100% tumor cell death. And we see the same phenomena that we always see, where at the initial time points, the tumor cells grow out, but then we're able to do the antigen assimilation phenomena and eliminate these tumors. When we add interferon gamma, we totally block that effect. So nothing happens in this. We're unable to kill the antigen negative cells with an interferon gamma blocker present in the experiment. When we look in the exact same experiment, but rather than looking at the antigen negative cells, we look at the antigen positive cells. We show that we have a very limited or null or small effect of blocking interferon gamma on killing the antigen positive cells. So this is really a phenomena that we're blocking the assimilation pathway rather than just knocking down T cell activity. So we were thrilled to see that data in the engineered models, and we wanted to test in a less engineered system. So here we found a cell line. This is another breast cancer cell line T47D that is null for ROR1 and PDL1 at baseline. And then we found another breast cancer cell line MDA MB231s that are positive for both ROR1 and PDL1. And the T47Ds, as basically every cell line we've ever looked at, upregulates PDL1 in response to interferon gamma. So we labeled one cell line in blue, one cell line in green, IRFP, BFP, and we were able to then show that in the monoculture assay, so when we have our antigen assimilation design and we're looking at killing the cells that are target negative, in a monoculture, we are unable to achieve any T cell killing. On the cells that are target positive, we have a very robust killing activity. And then very similarly to what we observed with the engineered cell lines, when we do the co-culture assay, we're able to kill rapidly the MDAs, which have the targets on them. And then once again, a delayed kinetic, once again, this sort of getting angry and then being killed, we see the same kinetic phenomena where on the target negative cells, we're able to turn the tide and eliminate those as well. So now we're showing this in a second model system. And then once again, finally, for this proof of concept design, we wanted to just see, are they active in vivo? We chose a preclinical prostate cancer model that we have experience with in the group. And we showed that these designs are totally effective in vivo, and we can totally eliminate this. We're looking at tumor volume in this in vivo study, and we're able to have durable and complete remission of the disease with this engager. So this was, there's lots of things from this proof of concept design that we think we can improve. And so that's really what I'll focus on in the next part of the talk. So once again, we're recognizing that solid tumors have a lot of barriers that we need to address. We're hopeful that with this antigen assimilation platform, we've addressed some of these involving heterogeneity, but we still have this issue of tumor selectivity. You can imagine that actually what we've built is a super toxic, strong drug now, because wherever it gets initial activity, it's going to spread and wipe out what's nearby. So now we think we need exquisite tumor selectivity to make sure that this is a safe therapy. So that's what I'll touch on next, is how our strategies for improving tumor selectivity. The question is, right now we're using ROR1 bivalent VHHs. There's a chance that would be selective enough for brain tumors, but we're also aware of some off-tumor, on-target normal tissue expression that could be a problem for the ROR1 target. So I just wanted, this is an idea or a strategy for improving selectivity that we first engineered in collaboration with Mignon Lo for targeting a unique rare leukemia, a mixed phenotype acute leukemia, where this is a cancer that has a mixture of myeloid and lymphoid lineage expression markers on its cell surface. So this is really a sort of a clear-cut case where there's lots of expression of CD33 in the body singly. There's lots of expression of CD19 on B cells in the body, but only on the leukemia are both CD19 and CD33 present at the same cell surface. And so the concept here is that by engineering affinity and avidity of our design, we can specifically anchor on and target dual antigen selective cells while having a very low, weak interaction with monovalent cells will not target the T cells to kill them. So this was a concept that we were thrilled that we were able to succeed or really achieve dramatic windows in potential therapeutic index. So we basically took CD19 binders, CD33 binders, we did a set of affinity tunings, and then we re-engineered them into the multispecific construct. And then we asked, after screening hundreds of these things and a significant amount of work, we were able to get to a lead design that has 100,000-fold and 400-fold difference in killing for dual antigen selective cells versus CD19 or CD33 alone. So the hope behind this design is that we can target the leukemia but leave intact healthy B cells and healthy myeloid cells so that these patients don't undergo, aren't prone to infections or other things that happen when you lose your B cells or myeloid cells. So this is the concept that we proved very nicely with this project, and now we're interested in bringing this into our antigen assimilating DMG targeting therapeutics. So this is work that's led by a postdoc in our lab, Ian Blumenthal, where he identified a set of promising brain tumor antigens that have failed in the past because of off-tumor toxicity. And the thought was these are well-validated binders, they're in the literature, there's drugs that are made, we know they're going to work, but the problem is toxicity. So he went and generated a set of affinity-tuned variants of each of these for several binders for each of these targets, and did so very successfully for ROR1, EGFR, and EpCAM. For targets B7H3 and GD2, unfortunately, we either got no effect on affinity or we knocked out the affinity when generating these. But now we have sort of this toolkit of a set of binders with tuned activity that we can employ in the same way we did for Impel. Some really cool things that Ian discovered with this work that we had anticipated but didn't expect it to be quite this clear were that affinity obviously matters. So here we're looking at a ROR1 binder, we're looking at a T cell killing assay and looking at differences in EC50. And what we see is that we have the wild type ROR1 binder versus an affinity variant, which we call V1. And we have, a lower affinity binder in this position A on this T cell engager. Really, there's a difference in activity that mirrors the difference in affinity. Something else Ian noticed, though, was that if you put the binder in position A on this heavy chain versus position B tucked off the light chain on the CD3 end, that it made a difference in the degree of activity. And then furthermore, that these two activities could synergize such that when we took the wild type binder in position A and the affinity tuned variant in position B, we now completely eliminated the ability of this design to direct activity against T cells. But it clearly still has some ability to bind and activate. So we think this is exactly the sort of thing that we're looking for, where we can affinity tune and now positionally tune the binders such that we can get this ideal dual antigen selectivity for the designs. So that's work that's ongoing, and I'm sure Ian will give a talk soon that he'll be able to update on the success of this approach. So another thing that I wanted to touch on with selectivity is that right now we're using PDL1 as the checkpoint blockade in the antigen assimilating targeting arm. There's a lot of studies ongoing in Jim's lab. Jim is advancing a PDL1 CD3 bispecific that's in very late stage development. And it's unclear at this point if there's toxicity associated with targeting PDL1 in the context of a T cell engager. In the chance that the normal expression of PDL1 may limit the therapeutic window that we're looking for, we're interested if there's other antigen assimilating targets beside PDL1 that we could use. So we know that when tumor cells or cells in general sense interferon gamma, they upregulate PDL1. We know they also upregulate MHC class 1 and 2 and other things. But the question is, are there other things that tumor cells do uniquely that immune cells or other normal cells may not? And surprisingly, we were unable to find a lot of literature on this. So we decided to just do it ourselves and find out, at least at the mRNA level, what genes are uniquely upregulated on brain tumor cells in response to interferon gamma that PBMCs don't upregulate. So we basically took pediatric brain tumor cell line, we treated with interferon gamma. We then did bulk RNA sequencing of that treated group at 0 and 48 hours. We also did the same thing for patients' PBMCs. We treated interferon gamma so that we get sort of this hallmark interferon gamma response. And then we did differential gene expression to see what we saw. And I won't go into the details of this today because it's a new data set and we're just now finding things in it. But what I will say is it's an absolute gold mine of new things. So we found novel cancer biology that happens here. So I jumped ahead a bit here. So yeah, what we found was comparing PBMC, what's upregulated on PBMCs by interferon gamma versus what's upregulated on brain tumors to interferon gamma in this one cell line. We found 300 genes that were roughly 300 genes that were overlapped, 800 unique to PBMCs, and almost 2,000 that were unique to the pediatric brain tumors. So there's a ton of crazy stuff that happens in tumor cells when they sense interferon gamma. Some of the things that we found are just, like I said, novel pathways, novel cancer biology. We found novel tumor-associated antigens, so things that we wouldn't have expected to be upregulated on tumor cells, we find, as well as we found novel druggable metabolic pathways that we think could synergize with our T-cell engagers. So I look forward to sharing some of this data in the coming year. We're also hopeful to do this for many more tumor cell types and really understand what's universal about these phenomena, what's unique to certain tumor types, and what's shared across, pediatric versus adult tumors. There's, I think, a lot to understand about this field of how tumor cells respond to immunotherapy. So I wanted to share this one approach that we're taking. Looking at the mRNA was very informative, but it's obviously missing a lot of things that could happen. So we're taking this additional approach where we're taking a pediatric brain tumor. We're going to treat it with basically start to kill it with a T-cell engager, so like a 24-hour T-cell killing assay. We're then going to harvest that tissue and inject it into a llama for a antibody binder discovery campaign. We also would love to do this with primary tissue, so growing these tumors in mouse brains, extracting the material, and then injecting that into the llama. And the goal here is that when we look at mRNA, we see genes that are upregulated. When we actually do an antibody discovery campaign agnostically, we're hopeful that we'll find novel VHHs or antibodies against things like maybe lipids or sugars, things like how GD2 was discovered, that uniquely respond by interferon gamma and are tumor specific. So this was another novel approach of this strategy. And this is work that Ian Blumenthal will be leading. Coming from a CAR background, we're really interested, could we use CAR T cells not as a therapy, but as a sensor for on-target, off-target, on-target activity. So the strategy, what we're going to do here is basically immunize the animals with treated brain tumors, take their VHH repertoire and clone it into a CAR library such that each cell in this CAR library will have a unique VHH on it. We will then incubate them with normal tissue sections. So anything in this CAR library with a fluorescent reporter, so anything in this CAR library that interacts with normal tissue will light up and we'll discard it. We'll then take the non-reactive population, put it onto tumor cells, and then select the population that binds or is activated by tumor cells, and then continually iteratively enrich that population with the hope that we find something agnostically that binds to interferon gamma treated or immunotherapy exposed tumor cells, but not to normal tissue. And then if we're lucky enough to do that, we'll then go under the difficult process of figuring out what it actually targets by mass spec or other things. But this is we're excited about this approach to try to find, truly perhaps a tumor selective, a truly, golden bullet tumor selective tumor antigen, as well as, I think, advanced novel CAR reporter strategies for binder discovery. Okay. So now we're trying to cross these challenges to solid tumors off one by one. The next one on the list is unnecessary and non-productive cytokine release, or CRS, in ICANS. So like I said, I think this is really the biggest problem that T-cell engagers face, is that you end up hitting CRS before you end up hitting efficacious doses. And so we're curious, how is it that, can we build better T-cell engagers that can increase that limit of CRS, therefore we can dose into efficacious ranges? And we identified that we think the CD3 arm of our bispecific is the best way to have to achieve that. So we were asking, can we improve biodistribution, serum half-life, unnecessary cytokine release, and therapeutic index with an improved CD3 binder? So one thing I'll mention that sort of prefaces this work is that in all of the FDA-approved bispecifics I showed at the beginning of the talk, almost all of them use the same CD3 binder. It's a human cyno cross-reactive. It's called SP34. It's one of the only human cyno monkey cross-reactive antibodies out there, but it comes with a lot of liabilities. In fact, the human cyno epitope that it interacts with is really negatively charged. And so just by definition, that binder has a lot of positive charge. And by that characteristic, it sticks to a lot of things that are negatively charged. So it's almost hard to avoid if you're going after that human cyno cross-reactive epitope that's negatively charged. Almost by definition, the SP34 binder and any other antibody you would discover against that epitope is going to have polyreactivity issues, which I'll show how big of a problem that truly is. So with Jim Olson, in collaboration, we launched what we called the end-all be-all leave no stone unturned CD3 campaign. We call it something else in-house, but I'll save that for the audience. And so we really wanted to hit this with it. We didn't want to fail with this campaign. It's a traditionally really tough target, and most of the things you get out are the antibodies that already exist, or you get a lot of antibodies out that don't activate T cells or don't bind actual cells. You get a lot of binders against the immunogen or soluble CD3 itself, but not against the actual T cell epitope of CD3. So knowing this, we took sort of a unique strategy where we first immunized with a holo TCR construct engineered to overexpress on rat C6 cells. So we did this campaign in partnership with OmniAb and their OmniRat platform, so that what we got out at the end were fully human, therapeutic ready antibodies. And so we engineered these rat cell lines that overexpressed human holo TCR, and that was what we primed the animals with, such that we were more or less hoping that we were guaranteed to get binders against the native context of CD3, which had plagued previous campaigns. Additionally, we knew that human cyno reactivity was really important. And so we designed these unique protein toroid immunogens that tease out, after establishing the memory response against the holo TCR, we then wanted to tease out the rare subset of those that were human cyno cross-reactive. So we built these unique defined asymmetric toroids, we call them, which I'll touch on in the next slides, to help really tease out those rare human cyno reactive clones. And just with this approach, we were really successful. We had 185 hits, 87 clones, 12 clonotypes, with a range of somewhat a range of native affinities. And then 7 out of these 12 clones bound human T cells very well, and also activated human T cells very well. So yeah, I just wanted, given the audience and the opportunity, to share a bit about our asymmetric or our toroid immunization platform. So a bit of an aside here, but this is the scaffold that I'm talking about. We call it a circular tandem repeat protein, or a CTERP. We also refer to them as toroids. This was something that was built by Phil Bradley at Fred Hutch, and looked at structurally by Barry Stoddard at Fred Hutch, and then functionalized in Jim's lab by scientist Colin Carenti when I was there as a postdoc. So what this basically is, it's a in silico design, so non-native protein sequence. It's a dihelical repeat. So there's 24 repeats that form this toroid. Each one is identical. And this can be used to present proteins on its surface. So sort of a unique scaffold where all presentation occurs on one interface. Unlike a strep tetramer, or unlike some of these nanoparticles which present things in all directions, this is sort of a directed presentation of antigens on a single face. What's also cool about this platform is because every single one is identical in a repeat, you can slice up protomers of it into any way you can slice a pizza, and we can create tetramers or trimers or octamers, all based on the number of repeat lengths, right? So if we have 6 repeats can fit around the circle, so we can make tetramers. If we have 8 repeats, that can fit 3 times, so we can make trimers, and so on and so forth. So just another way to describe this, this is or how we make these immunogens. This is a sort of at the genetic level. we have a basically genetic fusion of a protein antigen or what you plan to present on the toroid, some linker, and then a given number of CTERP repeats with an affinity tag for purification, such that we can build these into a mammalian expression vector, transduce a mammalian producer cell. That cell then secretes these individual protomers, which spontaneously self-assemble into the stable tetramer. So the first thing that we did as far as an immunogen or a vaccine with this platform was with Andy McGuire at Fred Hutch and Neil King from the IPD, where we took our toroid tetramer construct and really put it up in a boxing match against the 7-mer, 24-mer, and the 60-mer sort of Icosavax nanoparticle designs coming out of the IPD. And we were asking questions like, is there a difference in immunogenicity? Is there a difference in neutralizing titer? Is there a difference in stability or epitope presentation between these various ways to present immunogens to an immune system? I'll say definitely going into this, the general hypothesis was that the higher order valency would equal more immunogenicity and more neutralizing titer. And what I'll say is that that wasn't the case. So we built, these are candidate EBV vaccines. So we use the EBV glycoprotein heterodimer GHGL as the antigen. So we made single chain GHGL, and we compared monomer versus the toroid former versus these higher order scaffolds. And what I'm showing here is the neutralizing titer from these experiments. And what you can see is that in comparison of the monomer reagent versus all of the higher order species, the monomer is a terrible immunogen unless it's a really poor response. But once you get the former and all the other higher order scaffolds are really quite equal in terms of their ability to elicit strong, both, I'm showing neutralizing responses here, but also this general titer. So there wasn't really this profound difference in higher order valency equals more immunogenicity. Furthermore, what we saw was actually there was a benefit of the toroid former in that it was able to more uniformly present all epitopes to the immune system. So here, once again, we're looking at GHGL. These are known antibodies against that target, and they're sort of some bind scaffold distal and some bind scaffold proximal. And what we saw was that for the toroid, because it's smaller, it has more flexibility, inherent flexibility. We found that all of the antibodies bind similarly to this design. When you look at some of the higher order scaffolds like the 60-mer, things like this antibody CL59, which is scaffold proximal, doesn't bind as well. There's less epitope access to the construct in this context. And that just sort of makes a lot of sense when we model these things structurally, where you can look at, here's the tetramer versus the 60-mer, and here's an antibody, everything's to scale. You can imagine this is sort of like trying to get to the trunk of a tree in a forest. It's a lot harder when it's really dense versus with this more spread out flexible design. So just to share, we think that there's a lot of benefits of higher order multimeric display, but that a former really can achieve all the benefits without a lot of the problems you get with these higher order scaffolds. So we've now shown that this toroid immunogen has a high expression level in mammalian systems. It's compatible with RNA, DNA vaccines. It has a really simplified production and purification scheme. They're incredibly stable. We can get hundreds of mgs, almost a gram in some cases per liter with these designs. And generally speaking, the scaffold itself is lowly immunogenic. So then that brings me then to the how we advanced this work now, where we engineered asymmetry into the CTERP. So this is the parental scaffold here. And what I haven't mentioned yet is that the protomers that come together to form, in this case, a tetramer, requires a disulfide crosslink in order to really stabilize it. So without this stabilizing disulfide crosslink, the toroid doesn't actually form. It kind of samples the positions, but if you run something like size exclusion chromatography, without the disulfide, it will run in its monomeric form. So we sort of took advantage of this phenomena, and in our redesigns of the interfaces, we also included moving the disulfide bond to unique locations, to the inside, outside, bottom, inside, outside, inside. So like we sort of sampled these different points in the interface where we could put the disulfide bond. We were anticipating that they'd be able to sort of sample each other, but then only in the defined where the disulfide clicks into position would we truly stabilize the design. And this, we just basically modeled this to say, if you made a parental C-terp stochastically, where you just took 4 different protomers, mixed them together, what would you expect to get? Well, you'd expect an equal ratio of all the different possibilities. When we clicked in the disulfide at the various, unique positions, we found that 100% of the time we got the way that we defined it to be, right? So now we can basically define interface 1, 2, 3, and 4, or protomer 1, 2, 3, and 4. So this was a concept and an idea. When Ray and I designed these, we gave it about a 2% chance that this would actually work. And then we were absolutely thrilled when Ray came in and showed me this gel from our first expression proof of concept design, where we took ASIM1, ASIM2, ASIM3, ASIM4, and we tagged them with different length cargoes. So we used just a random VHH. So ASIM1 has no cargo. ASIM2 was fused to 1 VHH, 2 VHHs, and 3 VHHs, so that we could see a size separation when it was formed. And we were just, thrilled to see that when we ran SDS page of the supernatant, so we haven't even purified anything yet. This is just a supernatant secreted from cells, that we had this band of the, by SDS page, of the correct molecular weight for a full form tetramer. And that when we broke those disulfides by a reducing agent, that we got this nice 1-to-1-to-1 ratio of ASIM 4,3 to 1. And we verified that by size normalized densitometry. We've then also shown by SEC that indeed this thing runs at the right disulfide cross-linked weight, and that by negative stain EM run by Barry Stoddard at Fred Hutch, that our actual, we were able to visualize nice, stable, monodispersed constructs that more or less look exactly like the cartoon that we drew up to describe them. So then this is the rationale that we're using for these, for our Immunogen campaigns, of how we think we can engineer cross-reactive or cross-strain or multi-reactive B-cell responses where otherwise you wouldn't be able to. So once again, protein engineer, not an immunologist, and I've already enjoyed the discussions today where I now understand this process better than I did yesterday. But this is sort of the dumb cartoon that we put together from our mind of how this works. So you have a B-cell receptor. If it engages with an antigen, it sends an activation signal. Yeah. When we build our asymmetric C-terps with now defined antigen species, one like human, mouse, cyno, canine, whatever you would want, preclinical species of interest, if you have a B-cell that can only bind one of these antigens, so if it's only human reactive, then it's going to send this monomeric activation signal. We know what that looks like because we did it with GHGL. A monomeric activation signal is super weak and doesn't look great. You don't get antibodies against it. What we do know also is that when you get a tetrameric-like response, so if you have a B cell that can bind all 4 of the antigens, or basically a conserved epitope across all 4 antigens, then you're going to transduce this tetrameric-like response and generate a strong humoral response that mimics the tetramer. So with this asymmetric design, we believe that we can now force or engineer campaigns such that only the cross-reactive B cells will be strongly activated. And this could be a benefit if you want a binder against multiple preclinical animals so that you can advance your therapeutic through those species. And in cases where you don't even know there's a cross-reactive epitope, this can agnostically sort of figure out what it is and generate binders against it. So this is the idea behind the cross-species asymmetric C-terp. And that's exactly what we did for CD3, where, like I said, we basically primed the immune system against the cells. We then used human cyno design, or all 4 species, human cyno, canine, rat, or mouse design. And by being able to boost the immune system with this binder, I think it's the reason why in the campaign we came out with many human cyno cross-reactive binders. We also noticed by looking at titer, we did many different ways. We immunized with DNA, we immunized with just cells, we immunized with soluble protein. It was the animals that received this strategy that really had the highest, the only detectable human cyno cross-reactive titer in their sera before the B cell discovery. So we really, I think, proved the validity of this platform. We've also, Sinduja, a postdoc in Jim Olson's lab, has also used this technology to discover cross-reactive CRLF2 or TSLPR binders that she's advancing. And we now have 3 or 4 additional campaigns ongoing in camelids using this technology. But yeah, so that was just a bit of a side on the Immunogen technology. Now back to the primary story of the reason we did all of this work was so that we could get novel CD3 binders so that we could build best-in-class T-cell engagers. So I just wanted to real quickly, I'll go through this to show sort of the unique properties that we got out from the binders from this campaign. So like I said, one of the biggest things plaguing CD3 engagers is that everyone uses sp34 and that binder is polyreactive, meaning that it sticks to things it shouldn't. And this was a really simple experiment we did to prove that. This was done by Ken Brazel, where we basically just took a Raji B cell, which has no business being stained by a CD3 antibody. It doesn't have CD3 on its cell surface. And we just pushed the staining. We stained at 10 micrograms and 30 micrograms per mil. And what we're observing here is that we're just showing that sp34 antibody sticks to a cell that doesn't have CD3 at a pretty appreciable level. And we observed this even at 10 micrograms per mil, which is not that high of, a pretty high concentration, but not that high. What we were thrilled to see was that many of our novel CD3 clones discovered from the campaign didn't have that phenomena. We've now done this against other cells, hex cells, and others, and we don't observe any non-specificity with these clones. Also, I'm highlighting that on top of that, we have human cyno-reactive clones. So we were able to access obviously non-sp34 epitopes with this campaign and generate human cyno cross-reactive binders that weren't plagued by this historic positive charge patch that has been an issue. I'll just highlight also that we were really excited about one clone uniquely that could bind delta epsilon but not gamma epsilon. So I'm just showing here a super shift assay where we take soluble CD3 and soluble antibody, we mix them together and after they form a complex in a column chromatography. So you can see with sp34, just a positive control, these are at reasonable concentrations. It can bind both delta epsilon and gamma epsilon soluble CD3. With our unique clone that is delta epsilon selective, we're only able to form a complex with the delta epsilon heterodimer, but not the gamma epsilon heterodimer. So this is really a fundamentally different epitope. And then we now have a BLI instrument in the lab, and Ray was able to do this really cool experiment just looking at the affinities of our unique delta epsilon selective binder for human CD3 versus cyno CD3. And we were excited to see that we basically have identical affinities in the low digit nanomolar, so 8 and 6 nanomolar binders for both human and cyno CD3. And then lastly, I wanted to touch on this point that because we're delta epsilon selective, so this is a picture, cartoon of a TCR, you have alpha beta chain, then you have a epsilon delta heterodimer and a gamma epsilon heterodimer. Most CD3 binders are epsilon specific. So you can imagine that when you're in a T-cell engaging format, that when you bind a TCR, you're binding sort of on both sides. You have the opportunity to bind on both sides. And then you sort of have this ability to like mechanically rip that receptor apart or really transduce a strong mechanical trigger on it. Whereas if you only bind delta epsilon, you're binding once per complex. And that's really represents a more native type of activation, similar to what you would if you were in a native setting where you're a TCR engaging with an MHC complex. That's like a one-to-one stoichiometry of signal transduction. Whereas you can imagine with a T-cell engager that's epsilon specific, then like I said, you're sort of pulling this mechanically stronger than you would otherwise. So what we're really excited to see, and this is a reproducible phenomena that's coming out with this binder, is that when we build T-cell engagers with it, here we're just showing a CD19, CD3 design, that they have really similar activities between sp34 and SCRI-S6 in terms of T-cell mediated killing. But what we see very reproducibly is that per unit of killing, we have less cytokine activation, which we think is going to be really beneficial and allow us to dose higher and not hit that CRS while still maintaining efficacy. And then just to show this sort of in concept, we took tarlatamab that I described was the only FDA-approved drug T-cell engager for solid tumors. It's made with the sp34 CD3. And we showed that this drug, if you just put it in a well with PBMCs, so no tumor, no target, at high enough concentrations, it activates T cells, right? This is exactly the problem, and we think why a lot of these drugs are causing CRS. And this is not like unreasonable high concentrations. This is below the concentration of the tarlatamab Cmax. So the concentration of this drug in patients in a dish, it's able to non-specifically activate T cells, we think, because of its polyreactivity. To prove that, we took one of our SCRI, one of our CD3s that is polyreactive, and put it into tarlatamab, and we saw a very similar phenomena where it non-specifically activates T cells without target. When we took our SCRI6, this delta epsilon selective non-polyreactive human cyno selective clone, we did not, at the highest concentrations we could test, we did not see any non-specific activity. Once again, suggesting that this is going to be a cleaner, safer engager built with this CD3. There's lots and lots of data on this binder that I'm not showing. We have now in vivo confirmations of this as well. But just for this talk, just wanted to show that we were able to isolate a truly unique special CD3 binder that can uncouple killing from cytokine release, as well as human cynoreactive. So then I'm going to, I think, jump through this story real quick because we're almost out of time. But when we think about another giant issue with T-cell engagers is the immune dysfunction in the TME, where there's negative immune regulators. Even if you bring a T-cell there and everything is there, there's so many negative factors that they don't work. One way we think we can overcome that is by co-stimulation. This was a really beautiful proof of concept experiment from Ian in the lab, where we took tarlatamab and we added co-stimulation bispecifics on top of it. And so this is now what I was showing before, were all the serial killing assays that ended after 5 days. In this case, we didn't just end the assay, we added more tumor cells back in to ask, can these T cells in the well continue to kill over time? So you kill the tumor cells, and then we just keep on feeding you more and more. And what we found was that tarlatamab is able to do 3 rounds of killing before the T cells lose their, whether it be exhaustion or T cell health, they're no longer able to control tumor after multiple challenges. We tested the 4-1BB and the CD28. These are co-stimulatory bispecifics, and added them on top of the design. And what we found was that with 4-1BB and CD28, we significantly extended the health of the T cells. With CD28, I believe that we could have continued to do this assay indefinitely. I think Ian had to go on vacation, so we just decided to call it here. But it really shows this is, FDA approved drug here. We add co-stimulation on top of it, we can dramatically improve T cell health and serial killing. And then once again, the point of this, the safety, there have been attempts in the clinic to add CD28 on top of T cell engagers, and it's usually a cytokine disaster, or really it's a toxic disaster. But we think because we have a super clean CD3 that doesn't have this non-specificity, that we may be uniquely able to add co-stimulation safely. And that's shown in this non-specific assay, where here the dark gray line is the little bit of activity that I showed before with tarlatamab. Just it has this intrinsic ability to activate T cells independent of target. When you add a co-stimulatory drug on top of that, it goes through the roof, and you get like maximum activation levels. Once again, there's no tumor present here, so this is a bad thing. When we do this with our SCRI6 design, I'm showing that same thing here, where this is a little bit of activity in light blue. Once you add co-stim, you get this dark blue significant signal. When we compare it to our cleaner CD3, we don't observe any non-specific activation, even when adding a strong CD28 stimulation on top of it. And once again, this is in the absence of tumor cells. For the sake of time, I'm going to skip over this, but just say that we have a novel CD28 platform that not only can co-stimulate, but can link in and basically make a single molecule that's CD3, CD28, dual antigen targeting. But it's a single molecule that breaks apart in the right environments. We can show that we can make that complex, that it can improve T cell kinetics of T cell killing, and that it doesn't have any non-specific activation, and that we're currently we've been able to do some in vivo studies and show that here in blue is tarlatamab versus controls, and this is about what you see in the clinic with tarlatamab. It extends life about 3 to 4 months, which is really impressive for this disease. But when we add our linked-in SAM co-stim, we significantly improve survival. And once again, we think we could even make this better if we continue to dose. This was just a preliminary experiment. There's a lot going on here, and was surprising to me was that our SAM co-stim versus the non-co-stim, that non-SAM co-stim that doesn't link in, there's a dramatic difference. And this is not due to killing activity, I think, but due to safety. And so I think these animals in the orange line, they're having good tumor efficacy, but these animals are coming out not due to tumor burden, but to toxicity of adding co-stim on top of a primary CD3. And our SAM approach basically eliminates that non-specific risk. So then just to conclude, it's sort of an exciting time for us because we've sort of developed all of these synergistic technologies that are out now all converging to where we think we can really build this super design that takes into account a novel CD3, adding co-stimulation, maybe dual antigen selectivity with antigen assimilation, novel targets, that we're really going to be able to put forward a drug that has a really good chance of helping to cure some of these really devastating diseases. So yeah, with that, I'll thank my lab, Ray, Haley, Ian, and Rob, and everyone in Jim's lab. We still are very thankful for Jim for all of his help, and we work very closely with his team. I thank our other collaborators in our department, Mignon and Beth and others who we work closely with and have lots of exciting projects. And then folks in CIIT, Sean, Rich, and Adrian, we're very excited about the projects that we've launched with you and look forward to potentially even more projects after discussions that we've had this morning, and look forward to discussions this afternoon. So thank you.
All right. Are there any questions?
Great presentation. I'm also new to immunology. So you showed us a lot of stuff for CAR T cells, but I'm wondering how is this going to apply to gliomas? Because I don't believe there are any adaptive innate immune cells, right? Or adaptive immune cells.
I really should have put this slide up front. I don't have it in this deck, but there's really, I agree. But something that I think is an underappreciated phenomena is that we believe T-cell engagers can bind to T-cells in the periphery and traffic them to the site of the tumor across the blood-brain barrier into the brain. So Jim Olson's lab has some really compelling images where they implant brain tumors in immuno-incompetent animals, add labeled human T-cells and T-cell engager, and demonstrate that in this system, there's an intact blood-brain barrier because of lack of albumin staining in the brain, and that these fluorescent T-cells can just light up the brain. In about 4 or 5 days, you see all of the T-cells in the body traffic to the brain. And I agree with you absolutely until I saw some of this data. And it's hard to argue that this is potentially what's happening.
Great talk. So I have a quick question about the, so you have all these designs, some of them have FCs, most of them have FCs or like FC-like things. Can you speak a little bit to the tissue penetrance of those, and is that an issue, like the ones with the FCs versus like the not like the ones with bad half-lives that seem to penetrate better?
So this is a debate. Basically, every T-cell engager now has an FC domain or a related half-life extension attempt. My personal opinion is that we've seen, across the board, FC-less T-cell engagers are more potent and more effective in vitro. In pharma, there's an issue with that model because the amount of drug it takes to get a certain outcome costs a lot more to produce. And plus just the benefit of people having to carry around fanny packs for constant infusion. But so across the board, we observe that FC-less designs are more potent. Sean actually has, with a syngeneic molecule that we built, that's mouse CD19, mouse CD3, minus and plus mouse FC, that the FC-less design is able to more readily, at lower concentrations, eliminate B cells in all compartments, including spleen and lymph nodes, whereas at the same concentration, the FC containing design is unable to do so. And those are, in theory, effectorless silent FCs. So if I could, from a first principles, I think FC-less designs with then you controlling the dosing is the best way to go. But there's other practical reasons why an FC half-life extension domain is reasonable. There's a company, Harpoon Therapeutics, who has a VHH-based platform, and they built albumin binding domains. And for whatever reason, whether it's that or other reasons, those drugs have not currently been effective in the clinic. For the same reason I was saying how polyreactivity and stickiness is like a death sentence for these drugs, putting the albumin binding domain is the definition of stickiness. FC avoids that because we knock out the effector function, and it's only the FcRn that's left in, and that only happens inside the cell. So in theory, the FC designs, effectorless designs, are still clean.
It's maybe more of a comment, because the specific question about does it affect tissue penetrance, based on our in vivo models, we don't think that's the case, because we can actually show that the B cells in the tissue are being opsonized by the FC-containing drug. They're just not being killed in the same way. We don't really have a great mechanism for why the FC receptor is disrupting that, but I don't think it's just this concept of it's too big or it doesn't get in or et cetera.
Yeah.
It's definitely binding to the B cells, it's not killing.
That seems like something.
Agreed.
I suppose I now have to ask a question because I took the mic. I was going to ask about the CD3 campaign you did. Just, I think I know a little bit about immunology, but almost nothing about CD3 signaling. The fact that it bound one of the forms of CD3, was that known a priori? Like, was it a known feature of the biology that you should have something target X versus Y?
No. I think so. We
not known to you or not known to
nothing that we did that we immunized with would have biased delta epsilon over gamma epsilon.
Yeah, I get that, but I suppose was the fact that the clone that you selected as the best happened to bind delta versus epsilon, could that have been predicted? And if so, are there further iterations based on known features of CD3 biology?
There's not. My understanding is there wasn't much known about that. There's one other delta epsilon CD3 clone that is people are aware of, and that's a super low affinity binder. And it's been hard to disentangle the activity that they observe in their assays and in the clinic. Is it because of the delta epsilon selectivity, or is it because of their super low affinity?
Yeah.
This is the only and first that we're aware of high affinity delta epsilon engaging design. So I think we have some ongoing experiments in the next couple of weeks where we're going to we have affinity tune variants of this binder now. So we're going to directly ask, if you tune affinity, what's the ratio of cytokine to killing? And if you have delta epsilon versus epsilon specificity, what's the difference in per unit killing cytokine release? So I think for the first time, we'll be able to address that exact question.
Super cool.
I'll ask a question. So you showed data that the ROR1 T cell engager increased PDL1 expression.
Yes.
So T cells can obviously elicit multiple cytokines. Can you modulate your T cell engager to reduce the amount of interferon gamma production and favor other cytokines to distribute sort of like, increase PDL1 expression in tumors?
That's an excellent question. The best thing I can think of now that we can do is by choosing the right co-stimulatory domain. So something like CD28, for example, really favors IL-2 secretion over other CD3 alone or other co-stimulatory domains. So I think through choosing the right co-stimulatory domain, I had some talks this morning with folks that maybe there's new co-stimulatory domains that I'm not even aware of that we should be going after that may further, allow you to sort of define what's better. Is high IL-2 low interferon gamma, or high TNF and low IL-10, whatever the case may be. In general, what we observe with our delta epsilon selective binder is that it's low across the board. It's not like it's one over the other one. It's just in general, all of the T cell derived cytokines are lower per unit killing. With co-stimulation, for example, we now see that low, but IL-2 comes up a little bit more. So I think, perhaps through selective co-stimulatory domains, we may be able to do something like that.
Okay, Kate has a question. I will have to.
Hey, yeah. Can I ask a question here? I had a question about your PDL1 ROR1 trispecific, and specifically if you've used that molecule in vivo and looked at myeloid cell killing, and do you think in DMG that sort of killing of PDL1 expressing myeloid cells will be helpful or harmful?
Yeah, that's an excellent question. That's a question for Jim Olson. So he has really advanced data on a PDL1 CD3 alone bispecific, including some cynomolgus monkey studies. I think the question of myeloid cell killing my personal preference is that, yeah, it could probably be beneficial because, you know, those things produce negative cytokines. It's unclear to me if they can be effectively, you know, reprogrammed to be good players. So in my opinion, I think it would be fine to eliminate those tumor, you know, negative tumor myeloid cells. But yeah, I'm unable to answer the details of that question. It's excellent, excellent question though.
Take one more question.
Causes CRS. Do you know if it is there is specific immune cells that are more that are actually binding more than others in terms of
yeah? So we don't think it's a polyspecific interaction. It's more polyreactive. So that's just, I mean, we've seen similar non-target binding to Raji cells, Hex cells. I forget, basically anything we show, HUVEC cells. So I think it's a general positive charge, negative charge thing, not like there's B cell antigen X happens to also bind at a high level. It's more of a general stickiness.
One there. Thank you very much, Jason.